So we have adenine. Adenine will complement with what? Class.
Yes, sir.
Adenine will complement with thymine. This is something I just said now, okay? This is what I just explained now, that the complementary pairing is formed between purine and pyrimidine, where adenine form a hydrogen bond with thymine, and the hydrogen bond they form is 2 hydrogen bond, whereas cytosine form a 3 hydrogen bond with guanine. Is that clear? So the complementary base pairing is done in such a way that cytosine complement with guanine, while thymine complement with adenine. Is that clear? Is that clear?
Yes, sir. Yes, sir.
So now I'm asking you here, we have a tetranucleotide here. We have a nucleotide containing, we have a tetranucleotide here containing adenine, cytosine, guanine, and thymine. And the first one is adenine. While forming complementary base pairing with adenine, adenine complement with what?
With
Thymine, sir.
Yes, adenine complement with thymine, and then cytosine will complement with
Guanine.
Guanine complement with cytosine, and thymine complement with adenine. So just imagine another tetranucleotide by the side forming this complementary base pairing. So you are going to have a double strand of the tetranucleotide. So this explains that the DNA is a double strand, and we have seen how the double strand is formed between the nucleotides of the DNA, where we have an adenine binding with thymine and guanine with cytosine. Exactly. Now the other thing is, or the other problem.
Which other two? The time. It's 5 o'clock already.
Fatima, you're making noise. Now, the other property we talked about is that the DNA is it forms a helical structure. The first one we explained, it forms a double strand. The second one is a helical structure. Now, how does it form a helical structure? We've already explained the history, how the helical structure was even proposed by James Watson and Francis Crick in 1953, I think in the previous module. So we have the 2 polynucleotide strands in the DNA, and we say these polynucleotide strands are usually, who is making noise? These polynucleotides, they are antiparallel to each other. What does that mean, antiparallel to each other? What it means is, remember we talked about 5' end and the 3' end. So if we have the first nucleotide, as we explained, as adenine, and we have a thymine forming a complementary base pairing. So the thymine, or the strand in which the thymine is the post, is usually in parallel anti-parallel to the other strand. For example, when you have this strand as 5' to 3', then the other strand will be 3' to 5' end. So we said the two strands in the DNA are anti-parallel to each other. One is running 5' to 3', while the other one is running 3' to 5'. So when you have this nucleotide, polynucleotides as a double strand, usually the two strands twisted around each other to form a common, to have a common axis. So they are arranged in such a way that the more hydrophilic ribose form the backbone, whereas you have the peripheral surface made up of the hydrophilic ribose, while the more hydrophobic side form a kind of a stack of the nucleotide, the nitrogenous base inside. So the nitrogenous base are the ones that involve in the hydrogen bond with each other and as such holding the structure together. Is that clear? Class, are we making sense?
Yes, sir.
Are we making sense?
Yes, sir.
So we have explained why or how DNA form the double strand. What are the forces that are involved in holding the two strands together? How does the DNA become helical in structure? What are the arrangements of the components of the DNA in the helical structure with regard to the nature or solubility of the component in the DNA? We said we have a hydrophobic, which basically form the nitrogenous component, and then the hydrophilic, which basically form the ribose component. And of course, we know by the chemical nature of the ribose, we know that it is hydrophilic in nature. We talked about the properties of water. We talked about how water dissolves a chemical substance, or how water interacts with the other molecules. So we know, or we have seen the reason why we can say the ribose component of the DNA are partially hydrophilic, while the nitrogenous base are considered hydrophobic in nature. That is why we have the hydrophilic forming the backbone, and we're oriented at the peripheral surface of the molecule, while we have the hydrophobic part stuck inside the structure, forming the hydrogen bond with each other. Now, we've already explained this in the previous listing, where we said how this DNA helical structure or double helix is being wrapped around the histone to form what we call a nucleosome. And we said that how this nucleosome is also further compressed or coiled to form what we call a chromatin fiber. We have a chromatin fiber of 10 nanometer diameter. We have a supercoiling at 300 nanometer diameter. We have, at 30 nanometer diameter. We have a supercoiling at 300 nanometer diameter, which further condensed to form what we call the chromosome. And this is what we see in the metaphase inside the cell. So this is how the DNA, after being formed, it is compressed to form a nucleosome, and then further to form a chromatin fiber, and the chromatin fiber is compressed to form a chromosome. So now we talked about the RNA. Simple. In RNA, unlike in the in DNA. RNA does not form a double helix structure. Therefore, RNA is just a single strand. But you can have an RNA single strand can pull back on itself to form different structures. We don't need to talk about the structures, but basically they can fall on themselves, just like we've seen here from the image. You can have the complementary base pairing, but you have within a single strand. It is one strand that falls off on itself to form the double strand using conventional complementary base pairing. And we have a different type of RNA. Sorry. Have a different type of RNA, and each of these have its own functions. So RNA is basically the main genetic material found in viruses and some eukaryotic animals. They perform several functions. So we have a messenger RNA, heterogeneous RNA, transfer RNA, ribosomal RNA, small nuclear RNA, etc. In messenger RNA is involved in the transfer of genetic information from the genes to ribosomes for the synthesis of proteins. Heterogeneous RNA is more or less a precursor RNA. When we will explain how transcription takes place, and after transcription, we will see what type of RNA is formed and how does that RNA is being processed to form a different type of RNAs. And what are the functions of a different type of RNA? Just like we mentioned, the messenger RNA is involved in the synthesis of protein. We have where the information needed for the synthesis of which amino acids need to be joined with another is obtained. And we have a transfer RNA that involved in the transfer of the amino acid that need to be added to the growing peptide chain. We also have a ribosomal RNA. The ribosomal RNA basically gives the kind of a workbench for the protein synthesis. It is formed like the workbench where the amino acids will be taken and then the formation of the peptide bond. And then we have small nuclear RNA that involved in the messenger RNA processing. We have a cytoplasmic RNA. We have a transfer messenger RNA. So all these are also classes of RNA which we would have talked about. Okay, difference between RNA and the DNA. From the previous discussion, while talking about the structure of the chemistry of nucleotides, we talked about the differences with regard to bases and with regard to the sugars and the structural conformations, which we just mentioned that DNA are double strand, while the RNA are single strand. While with regard to sugars, we say that in DNA, what we have is deoxy sugars instead of the, we have deoxyribose sugar instead of the ribose sugar. While with regard to bases, we have guanine, adenine, thymine, and cytosine, forming the bases found in DNA. Whereas the bases found in RNA, we have adenine, guanine, cytosine, and uracil instead of thymine. So we have seen the difference with respect to the base composition of the bases. We have seen the difference with regard to the nature or chemical nature of the sugar. The ribose sugar, one is ribose, one is deoxyribose. With respect to the structural conformation, we said the RNA exists as single strand, while the DNA exists as double strand. And with respect to functions, in each case, we talked about the storage of information, the storage of genetic information in all the cases, but we have we have a different functions of RNA, we've seen how different type of RNA perform different functions apart from the fact that they also form genetic material in viruses like HIV virus. And we also seen how they play important as the in other eukaryotes. Sorry, my attention is being taken by. Are we together, class? Come, please.
Yes.
Are we together?
Yes, sir.
Okay.
Yes, sir. Yes, sir.
The punctuation synthetic nucleotides. Remember, we talked about the functions of nucleotides in our previous class. Do you remember? We talked about several functions apart from the fact that nucleotides form the building block of DNA and RNA. We mentioned about 5 to 6 of the functions of these nucleotides. Now, this nucleotide can also be synthesized. And one of the functions, they are used as either anti-cancer agents or antiviral agent. Why are they used, for example, as anti-cancer agent? You can have drugs, anti-cancer drugs, that are used for the treatment of cancer. But they are basically nucleotides, not exactly nucleotides, but they are synthesized to look like a nucleotide, not exactly nucleotide. Why? Simply because, for example, what happened in the case of cancer? In the case of cancer, we have uncontrolled cell division, where we have several cells, where we have a cells divide uncontrolled to produce multiple copies, right? Before cells,
Yes, sir.
before cells divide, there must be a synthesis of DNA. One of the first steps in cell division is the synthesis of genetic material. The genetic material needs to be duplicated so that you have one cell will have a two nucleus, so that the nucleus will separate, and then you have the separation of the cytoplasm, which we call cytokinesis, so that you have two different types of cells or two daughter cells, and each of these daughter cells has its own nucleus. So the first stage in the cell division is the synthesis of genetic material. And for the genetic material to be synthesized, what happens is just to copy the genetic material from the mother cells. How do you copy the genetic material? You use the nucleotides. You just have, remember we talked about the double strand of the DNA. So that double strand will open, that formation of the complementary base pairing will open. After it's open, then a new DNA will be synthesized from all the two strands. How is it synthesized? It is synthesized using the nucleotides. So the nucleotides will come and they keep on forming a base pairing, until you have a lengthy part of the DNA being copied. So that was copied will be released, and then the copy will continue until the whole DNA is copied. So when you have this nucleotide forming a complementary base pairing continuously like this, when you have an individual who is taking these synthetic nucleotides, these synthetic nucleotides will go and bind. It will also bind just like the other nucleotides bind. But being the fact that this nucleotide is not like the other nucleotides, when it binds, it will stop the enzyme, because when it binds, the enzyme will pick the nucleotide, go and attach it. Another will come, go and attach it. another will come, go and attach it. But once this synthetic nucleotide come, it will bind to the active site of the enzyme. We'll talk about the enzyme, we'll talk about the active site. We know what I'm talking about. So once that enzyme comes and binds to the active site of these synthetic nucleotides, the synthetic nucleotide will bind to the enzyme and will not release the enzyme. Therefore, even the enzyme will try to incorporate it to the DNA. It will incorporate, but it will not release. Therefore, the enzyme will not be available for another nucleotide to come and to continue adding the nucleotide. As such, it inhibits the process. Inhibiting the process stopped the process of DNA synthesis. Stopping the process of DNA synthesis will stop cell division. And that is how these drugs kill or stop the cancer cells from growing. Is that clear? Class, is that clear?
Yes, sir.
Fatima. Fatima, are you with us? Fatima is not with us. Jen. Jen, are you with us? Bisola.
I'm with you, sir.
Good. Fatima, you're not with us.
I'm here, sir.
Please. Did you hear what I just said?
No, sir. I was attending to a patient, so I had to move and just attend to that patient. Sorry, sir.
So basically what I'm saying, what is the mechanism of synthetic nucleotide as anti-cancer agents? Why synthetic nucleotides are used as anti-cancer agents? For those of you that know some drugs used against cancer, maybe you will check, when you check the leaflet, you will, some of them, you will find out that they are synthetic nucleotides. They look exactly like these nucleotides we are talking about, only that they are a bit different. And the difference make them to be a bit more reactive than the normal nucleotides. So what happened? What exactly happened is in the cancer cells, there is uncontrolled cell division. And for this uncontrolled cell division to take place, there must be a continuous synthesis of DNA, because for cell to divide, there must be a copy of DNA, of genetic material. Because when cell is divided, the cell make a duplicate copy of everything. For example, the subcellular component, it make a duplicate copy of ribosome, it make a duplicate copy of all these cellular organelles, including the nucleus. So for the cell to make as a duplicate copy of the nucleus, then the nucleus contains DNA. Therefore, it will make a duplicate copy of DNA. And what is the enzyme that make a duplicate copy of the DNA is what we call DNA polymerase. So DNA polymerase is responsible in using this nucleotide to synthesize the DNA. So when DNA polymerase is synthesizing DNA using the nucleotides, when you have an individual or a patient taking a synthetic nucleotide as anti-cancer agents, this DNA polymerase will pick the anti-cancer agents, thinking that it is the normal nucleotide in the system. So by taking it, it will now inhibit the enzyme from doing its function, thereby inhibiting the process of DNA synthesis. Stopping the DNA synthesis will stop the duplication of DNA. Stopping the duplication of DNA will definitely stop the cell division. Stopping cell division will stop the cancer cells from dividing. Are we together? Class, are we together?
Yes, sir.
I hope we're
Yes, sir.
Are we
Yes, sir.
listening? So this is basically the mechanism through which synthetic nucleotides they served as anti-cancer agent, and the same thing happened in the case of antiviral. So this one, I don't know. Okay. So the next thing we talked about is we're going to talk about DNA replication, transcription, and translation. And before we talked about that, we need to tell what is the central dogma, because the three put in one picture, we call them a central dogma. In central dogma, we are explaining how an information in the DNA is being utilized or being copied from the DNA to form protein. Remember, we said DNA is a repository of information. Information on almost everything. When that information is needed, how do you collect the information? So this information is collected or obtained from the DNA through this process we call transcription and translation. But we will also, in addition, talk about another process we call replication, how this DNA or a copy of this DNA is formed. Now, talking about replication, something I just try to explain while explaining the mechanism of action of anti-cancer agents. Replication, defining replication is a process by which a copy of DNA is formed by the action of an enzyme called DNA polymerase. Why is the copy of DNA formed? Why do cells want to make a copy of DNA? A copy of DNA is formed usually during cell division, where you have a complete genetic material is produced. When you want to form a new cell, new genetic material must be produced for that cell, just as I explained. And that process of cell division is what we call mitosis. How does a copy of DNA formed, or sorry, of genetic material formed, or a DNA is formed? Remember, I told you the DNA, number one, is a double strand in form of helical structure, and the helical structure is wounded, coiled around some proteins we call histones, and this spores are coiled, wrapped to form what you call the chromatin, and the chromatin also further condensed to form a chromosome. So if a copy of DNA is to be made, all this wounding and compression condensation is being relieved so that you will have the DNA, including the helical structure, will be relieved so that you will have the DNA as a double strand. So when you obtain the DNA as a double strand, that particular, you open it, so the complementary base pairing itself will also open. When the complementary base pairing is open, you have the single strand of the DNA, just as we have in this picture. So first of all, we have an enzyme called helicase that will come and open the DNA. It will remove the complementary base pairing, the hydrogen bond and the hydrogen bond between the bases. It will open up by an action of an enzyme called helicase. some protein, we call them single strand binding proteins, will stabilize the opening so that it will not go back, because they can easily go back and form the double strand. The next thing you have, the DNA polymerase will bind. You can see the enzyme. If you can see my cursor here, you can see the enzyme. Or is my cursor? You can see the enzyme here, DNA polymerase, is now adding a nucleotide to forming a new DNA strand here, polynucleotide strand. And you have this DNA as the newly formed DNA. And you have the other DNA also, you have a DNA polymerase coming to synthesize another DNA. So the basic things is, I'm not trying to tell you all this, the stepwise how this is formed, the formation of what you call a Kozaki program and the rest. The most important thing is that this is how DNA is formed. The process is called DNA replication. The enzyme involved is DNA polymerase. And the process involves the unwinding of the DNA, opening of the DNA helical structure, and the double strand is opened, and you have the DNA polymerase will bind, and the DNA polymerase will synthesize the new DNA. This is basically the DNA replication. Is that clear? Class, is that clear?
Yes, sir.
Yes, sir.
Now, the next process is called DNA transcription. And what is transcription? Transcription is the process by which the genetic information in the DNA is copied. It's copied to form RNA. The RNA that is just formed is called heterogeneous RNA. So depending on what are you going to do with the information, is the information going to be used to synthesize a protein? If it is the information, if the information is going to be used to synthesize a protein, a particular type of enzyme called RNA polymerase II will be used, and it will produce what we call heterogeneous RNA. That heterogeneous RNA will be converted to what we call messenger RNA. So the messenger RNA is used for the synthesis of protein. While if a ribosomal RNA is needed, so the information to be copied will be copied by a type of RNA polymerase called RNA polymerase I. So RNA polymerase I will produce heterogeneous RNA. The heterogeneous RNA will be processed to form ribosomal RNA. And remember, we already mentioned the function of ribosomal RNA. Ribosomal RNA form a kind of workbench for protein synthesis also, because all the three RNAs, the three here, they are involved in RNA synthesis. Only the messenger RNA contain the information, while the other ones, they only play important role in the synthesis. So you have RNA polymerase III also that formed heterogeneous RNA, which later used for the synthesis of transfer RNA. Is that clear?
Yes, sir.
Sir, please, can you re-explain this? Sorry, sir.
Sorry? Explain this again.
Yes, the transcription, yes, sir.
So this is transcription, and we say transcription is the process by which information in the DNA is copied to form, is copied to either form protein or to form the RNA, depending on what is needed. Because this information can be copied to form RNA or can be copied to form the protein, depending on what is needed. If the protein is need to be formed, we have a particular type of RNA polymerase that will copy this information from the DNA. Just as we explained the replication, this is exactly what happened. You have the DNA open, and instead of the DNA polymerase, you have the RNA polymerase will come and bind. So the RNA polymerase will bind and synthesize the RNA instead of DNA. You remember, DNA polymerase synthesize DNA. So when it binds, it synthesizes DNA. While when the RNA polymerase binds, it synthesizes RNA. And what it synthesizes, whether it is RNA polymerase 1 or 2 or 3, it synthesizes what we call heterogeneous RNA. Before the heterogeneous RNA, if it is RNA polymerase 2, it will be processed to form a messenger RNA. So how does even the synthesis happen here? Remember, we talked about complementary base pairing, and how does the DNA polymerase synthesize the DNA, where when you have this open and the DNA polymerase come and bind, and the first nucleotide, for example, say it is adenine, the DNA polymerase will add a complementary base pair, which is what? It will add thymine. So the DNA polymerase will shift to the next nucleotide. If the next nucleotide is cytosine, the DNA polymerase will add guanine, like this, until all the nucleotides will be added and synthesized. But if it is an RNA polymerase, when RNA polymerase comes and bind, for example, if it bind on adenine, it will add what? RNA polymerase will add what? Class, are we together?
Yes, sir.
If RNA polymerase bind, for example, it binds to adenine, it will add what?
Is it cytosine? I'm not sure.
In complementary base pairing, I just mentioned complementary base pairing. I explained how complementary base pairing happens and what nucleotide bind to which nucleotide. Fatima. Jen. Mariam.
I'm here, sir.
Now I'm asking, you have an RNA polymerase. Okay, you have a DNA polymerase. Remember, just listen to me. When you have a DNA polymerase binding to the orphan DNA, it will synthesize the DNA in such that the first nucleotide on the DNA that was opened is adenine. Based on complementary base pairing, the DNA polymerase will attach a nucleotide that complements with adenine. And that nucleotide is what? We said it is thymine. So anywhere the DNA polymerase read adenine, it will complement it with thymine. So they will form a hydrogen bond. So the DNA polymerase will shift. When it shifts, the next nucleotide, for example, the next nucleotide is thymine. So it will bring adenine and form the complementary base pairing. Like this, it will continue. If it is cytosine, it will bring guanine. Move next. Next, it is guanine. It will bring cytosine. Move next, it is adenine. It will bring thymine. Like this, like this, like this. Are we together?
Okay, sir.
So in the case of RNA polymerase, if it is RNA polymerase, remember RNA polymerase will not synthesize DNA, right? It will synthesize RNA.
RNA.
Yes, so if it is synthesizing RNA, it is synthesizing RNA from DNA. So once you have the RNA polymerase bind to the DNA, for example, the first nucleotide says is guanine. The RNA polymerase will add cytosine.
Cytosine, okay.
When it moves and it is cytosine, it will add guanine. When it moves, it is adenine, it will add what?
Thymine.
Are you sure it will add thymine?